So we all ask ourselves this question, quietly or out loud. Is there any hope for treatment? And I hope that today, as the sessions progress, the answer is starting to be more optimistic. The next talk will give you a glimpse into how that hope turns into a process and eventually into possibility and reality. We're so thrilled to welcome Richard Novak, CEO of Unravel Bio, a biotech company developing therapies for rare diseases. Earlier this year, with all your participation and your funding, our foundation awarded funding to Unravel to explore potential treatment strategies for ZTTK using their novel AI drug prediction and screening platform. Please join me in welcoming Richard Novak.
Good afternoon, everyone. It's really just wonderful to be here, and I've just enjoyed having the opportunity to meet many of you over the last, today and then a little bit yesterday. So I'm Richard Novak. Thank you for the kind introduction, Ada. I'm here from Unravel Biosciences. We're actually located a couple miles north of here, so we're in the Boston area. And I'll share with you first sort of how we think about diseases in a very different way, and especially aimed at therapeutics. And then the latter part of the presentation is really your data. So as Ada mentioned, we've been able to work with 22 families, so 22 sunshines and 22 healthy individuals, within that family for a really interesting initial study. And we have some initial ideas that could actually translate into therapies. So my disclosures, I'm CEO of Unravel Biosciences. Hopefully that's an obvious disclosure. So this is something that I really shouldn't be the one talking about. You are more experts on this than I am, but I think just to get everyone on the same page, there's a very unpredictable diagnostic odyssey, and many of the families that I spoke with today have shared theirs. None of them were easy. So of course, just to go over this, right, the idea is that these first symptoms, something, usually as a mother, like something's off, right? And so you go through this lovely, very complex path, usually ends up with some symptomatic treatment. Dr. Google comes in there somewhere. And then, more and more people are getting genetic testing, even as a first-line attempt at figuring this out. You can have a diagnosis, sometimes relatively quickly, sometimes there's no diagnosis. So since it's after lunch, and just to wake everybody up, I want to see your hands if you think 100% of patients get a genetic diagnosis with genetic testing. Good. No one, yes. 80%, 60%, 40%, 20%, 10%. Okay. So it's around 60% to 70% do not end up with a diagnosis. So do not, right? So the 30 to 40% do nowadays, and that's really kind of throwing the book at every possible diagnostic you could do. So hooray, you have a diagnosis. That means successful treatment, right? Well, sometimes, not in the case of ZTTK, because there's nothing approved. And if there's nothing approved, well, you have this diagnosis, but now it really hasn't helped you figure out the treatment side. And that's about 95% of patients we heard today, right, earlier in this morning. So this is just the diagnostic odyssey, right? But then I think what all of you are looking at is, well, what about the therapies? It's nice to have a name for it, but it would be even better to do something about it. And so the whole idea is, all right, how do we tackle that 95%, right? And even for the 5% that do have treatment, just because something is approved does not mean it actually works for any one patient. And anyone with a non-rare disease has encountered, oh, this works for most patients. Of course, you end up being like the statistical outlier in all those cases. At least happens to me all the time. And so what we did at Unravel is kind of rethink this whole process. So traditionally, you would start with some sort of target, and you heard about this a little bit in the morning, some rationale, some scientific basis for why you think a treatment should work. And then you go into animal models, cell models, preclinical models more generally, and then you go into patients. The success of that is On average, across all clinical programs, 2%. So out of 50 attempts, 1 will be successful, right? So that's not great. And so we were thinking, how do we make this more efficient? And so we started working with patients, because ultimately it's the patients that respond to treatments rather, not the diseases. And this is William Osler, who actually was considered the father of modern medicine. Early 1800s, he coined this phrase, right? That the good physician treats the disease. The great physician treats the patient who has the disease. And as all of you know, people are different. The patients are different. I mean, just the ones that are in this room and in the next door room, you can tell that not everyone's the same. And so what is a patient? And this really gets at how we work, right? So a patient, this kind of amorphous concept, is the sum of the genetics, not just the gene of interest, not just the SON gene. That does not define who a patient is. And you'll see what I mean later. You have 25,000 other genes. They're doing something, good or bad, neutral. They're in there. Environment, right? So some of you came from really far away. You might have jet lag. That's impacting how you're behaving today, like it or not. Medications, what you ate a week ago probably has some effect. And then time. Patients change. We all know that, right? And we need to incorporate that into the therapies. And so what we've been doing is really working almost exclusively with rare disease. And the whole idea is that we partner with patients, because that's the problem that we're trying to solve, not some amorphous disease concept. And one thing that's really unique about us is we find existing drugs to ask a very important question that I know you all care about, which is, does it actually work in this patient, that patient, right? And you can do that with existing drugs, because while we as a biotech company want to build new molecules that are more tailor-made, more efficient, maybe safer, maybe just better for the patients long term, that might take you 10 or more years, maybe $1 billion. And you remember, you have this 1 in 50 success rate when you do that. So that's not good. So is there a way to do this faster? And so we said, let's use these existing drugs. They may not be perfect, right? They may have other downsides, but the important part is clinical validation. Does it work in an actual patient? If yes, that's everything. If not, I couldn't care less about it then. And then we want to help patients today. So treatments today are worth something, right? You may want to wait for 10 years for the ideal drug that may come, may not, but what can you do today? And so that was really the idea here. And so with the advent of high-power computation, AI, we built a very unique platform, and it's called BioNav, like a bio-navigation system, and there's going to be a navigation analogy coming up. And the whole idea, and some of you have actually participated in the study that I'll describe in the latter part of this presentation, we can essentially do N of 1, like just patient by patient at a time, essentially a clinical trial in a computer on 40,000 molecules. Now, most of those you could never give to a person in real life, but we were able to actually ask, what would you get if you gave a person aspirin, or a really toxic cancer drug, or anything in between, right? And just let's computationally look at what the outcomes are. And so this is actually a real picture of the underlying network that we built for health. This is what health looks like, I guess. But then we can act on that with each of the 40,000 molecules, and it's hundreds of millions of different paths that you can take. But with computers, you can just ask all the questions at once and just score the results. And so how do we do this? Well, this relies on RNA. And so I'm really, really glad I wasn't going early this morning, because you had some really good baseline biology education. And so RNA, as you remember, right, there's the DNA, which is sort of the blueprint, the recipes for your cooking. The RNA is sort of, what do you want to do with it right now? Which meal do you want to prepare out of this whole cookbook? And then that makes the proteins. That's your meal, right? The DNA is static. Like if you have a cookbook on a shelf, that's not changing, right? But you might decide to do a fancy dinner, or pancakes for breakfast. You might use the cookbook in a very different way. All of that is then captured by the RNA. And so DNA is what all the sunshines were diagnosed with, because it doesn't change, and that's great for diagnosis. The problem is it doesn't change. So as your sunshines grow, as they maybe take medications, maybe respond in different ways, the DNA isn't changing. It tells you nothing about what may or may not be happening. And that's where RNA comes in. And so things like diet, microbiome, traveling from abroad to Boston, messing up sleep, all of that will affect it. And so another analogy, I know you've been bombarded by analogies, but here's a different way of thinking about how we work, which is Google Maps. So my guess is that everyone here either directly use Google Maps or some mapping software, or their Uber driver used Google Maps or some mapping software to find their way here. Because ultimately what you care about is you go from wherever you are now to your destination. You kind of don't care how you go there, just as long as it gets you there in a reasonable amount of time and money. And so that's what we developed with BioNav for patients, right? So in this example, we have a disease state, and that's your sunshine. And so by using what I'll show in a moment, nasal swabs to collect the RNA, we can define in this health map where that sunshine is, and we can compare them to their family control, right? And so now it's sort of like Google Maps. You have a starting point, which is the sunshine, and you have a destination, which is the, let's say, parent or sibling. And then what we can do is we can just ask, what are all the possible ways to go from your starting point to your destination? And this is like how Google Maps works. And so if there's, let's say, a traffic jam on your usual route, you go around it, right? And so we can now start looking at all those paths, which are molecules. And so it's not just a theoretical concept, but this is something you could actually bring to the clinic. And so that's what I'll be focusing on the rest of the talk. What we do as a company is then figure out, okay, now we know something works. We can then turn that into targets that we know work, and then into even better molecules. This takes time, this takes a lot of money, this is your biotech development program. But it all starts with all of you, it starts with the patients. Like, what are we looking at? What problem are we trying to solve? And so that's where the rest of the presentation will focus on. And the whole idea here is it's really personalized medicine, and it's making medicine not just personalized, but actually predictable, right? So it's not about, you have some chance of this happening. It's like, okay, but these are the therapies you could use to actually fix the problem, whatever that problem is for those patients. And so what we developed over the last few years is a way for patients to work with us without having to go anywhere. So we ship these nasal swabs to their homes. We've got some families in Europe and other parts of the world that participated in the study, not just the US. Mail the sample back. That lets us basically get insights into each patient and the control, equally important. We analyze it using BioNav, and then we get all these different AI-enabled outputs. What's going on with this patient? And it's, by the way, we don't put in the diagnosis at all. It's just the RNA that we feed in. We can find new ways of treating it. We can find potentially paths that clinicians could explore for treatment. And it's really this personalized medicine path. And you may ask yourself, well, what can you get out of the nose? What can you learn from that? And it turns out quite a lot. And this was actually surprising to us as well. But it turns out when you look at everything from this network Google Maps perspective, it turns out you don't really care about where the tissue is. So you don't have to get a brain sample if you're interested in brain function. Turns out people don't like to volunteer brain tissue. So the whole idea was, I can't imagine why. So the whole idea is, well, how can you make this accessible, right? Again, it's all about partnering with the families and with the patients and not add any more than you have to your lives. And so we found that it's basically a COVID and other respiratory pathogen kit. But what's important, the swab is just a regular swab that I'm sure all of you have taken at some point. But it comes with a very special preservation solution that does two things. It inactivates any pathogens. We actually don't want the viruses. We don't want the bacteria. We don't want those. We want the human cells. And it stabilizes it at room temperature. So you don't have to freeze it. It can sit in customs for a week, and we don't care. It doesn't affect the quality. And so now we can ship anywhere in the world, minus a couple countries. And yes, it tells us a lot about your brain. Most of the genes, or basically all the genes that are in your brain, show up in the swabs. And all these pathways that may be changing and be causing cognitive problems, and motor function, walking, all these kinds of things are represented. So it's a very interesting source of data. And again, I can't emphasize this enough, especially for rare disease. What we're working on is making rare disease more unified, and part of that is geographic. It turns out these mutations happen all over the world, like it or not, and so let's figure out how we can work with patients. I mean, one of the most challenging ones was the outback of Australia. It took them hours to find somebody who has a truck who could go several hours to drop off the envelope, and yet, they're part of the study, right? Like that's the amazing part. And so, we work with with many, many patients all around the world. I always joke that there's no penguins yet, but we have worked with sea lions, actually, so it's a fun aside. But then the question, of course, that you all wonder is, okay, well, does this work? Does this have any promise? And so I want to walk you through 3 examples, so 2 on this slide and 1 on the next. And so all 3 families work with us, in this case, individually. But the whole idea was these are extremely, extremely severe patients, and they're running out of options, effectively. And that's usually the types of families that work with us individually. And so first one is a boy with Rett syndrome, and usually this is considered a girl's disease because it's mostly little girls who develop it, and that's because it's so severe in boys. And so this boy was not doing very well, was in palliative care, and our main program commercially is actually in Rett syndrome, and so they contacted us and we did this work. Turns out we did this nasal swab analysis, and we found that the same drug that we're developing, and we're about to actually start 2 clinical trials with, would be predicted for this boy. So again, from a personalized standpoint, he would be part of that group that we'd anticipate to respond. The clinician, based on that data, based on mouse data that we already had, prescribed the drug, and they saw improved sleep quality, normalized blood metrics, very objective measures of some of the disorder that he has, and importantly, cognitive function and awareness and some motor function was actually dramatically improved. So that was really exciting. On the right side, very different disorder. It's called KMT2B dystonia. This boy actually has very little dystonia, which is basically sort of like very stiff muscles, but a lot of cognitive problems. And sleep problems, basically sleeping maybe 1 hour a night, something extremely severe and affecting not only him, but the whole family. And so we found an existing drug, it's just anonymized as Ravel-68, and the clinician actually prescribed it before we even had a chance to discuss the results. And then sleep was basically perfectly normalized within 2 weeks, and he started recovering receptive speech, so understanding what his parents were telling him. And a few months later was actually even able to start using some sign language to communicate back. Interestingly enough, he actually went through a really bad behavior phase because he got frustrated. He realized he was able to communicate mentally, but unable to do it physically. And so they actually had to teach him how to communicate again. He lost all those abilities. And so what's really exciting, I can't stress enough the importance of these partnerships because these are sort of N of 1 studies, but it doesn't stop there. So on the left, there are 2 more patients who saw the results of this work, again, very severe cases, who are about to start on vorinostat. I mean, this is an oncology drug, right? So people don't do this lightly. And then that data enabled us actually to file some clinical trial applications that are enabling even bigger, more statistically robust studies. On the right, there are 2 families that we work with specifically. They started a foundation. They didn't have a foundation. These patients were not connected. They didn't have Nathan and Ada. So they became Nathan and Ada for this community. And you literally just started this, or working with Combined Brain, and working with them for an observational study. So taking this drug, and there was another drug that a different family used that also benefited this little girl. They're starting their first observational study. So their first project is actually a clinical study. And that's what this enabled. Again, thanks to their partnership. And then I want to share this story with you. So this is actually a very severe patient with Cornelia de Lange syndrome caused by the SMC1A gene. And it's very prone to seizures as a syndrome. She lost the ability to walk after a really bad status epilepticus, extremely severe seizures. And she was basically declining over about 18 months. We worked with her about 6 months into that. And she spent over 100 days in the hospital in a given year, right? So extremely severe. And they sort of sat on the results because at some point they basically said, with their clinician, we don't want to do any more potential risk, any more harm. But she was getting transitioning from palliative to hospice. And they're like, okay, well, let's go back to those results. And so they did, and they ran some tests that showed she was actually deficient in two molecules, and they administered very high doses of those. And now she can walk. And she's, again, not cured, right? So this is not a magic treatment or anything, but she can walk, she can communicate however she was able to before, and basically it kind of addressed the issue that she started out with 18 months ago. So it didn't address the disease overall, right? It's not a magic bullet, I can't stress that enough, but dramatic improvement in quality of life, not going to hospice, and is now being, we had a video call with the family and she was part of the video call, right? So very, very strong impact, and there's about 7 weeks of treatment, so relatively fast as well. And so these are just 3 examples. We have several others, some that I can't, don't have the permission to share. But what we're working on really is kind of a different business model, right? So it's partnering with families through our program called RareShift, and that's really how we've been working with ZTTK, with the foundation. And this is rapid drug repurposing, right? Out of the tools that are available today to clinicians, what could help you? Right? That's that question. On the right, that's how we actually started the company as a biotech. We want to make drugs that actually truly address this disorder, right? And that can take time. The ideal molecules almost certainly do not exist today, so we have to make them. This takes a lot of time, a lot of money, but we can start with the clinical validation through patient partnerships that can help the patients today, at least partially, to be a stepping stone towards these better drugs, right? And again, this might take 10 years. So I want you to be realistic about this, but at the same time, there is promise along this path. You don't have to wait for the perfect solution to have some benefits in the process. And so we work with over 70 different disorders. This is, we managed to scale this up. We add other patients like once or twice a week easily. But what's really interesting to see, we start with the BioNav screen, and in many cases it goes through, let's say, an animal model. We actually have tadpole models that we can make. But the idea is to get into the clinic as fast as we can, and then we go back and then kind of fill in the gaps that you'd want to fill in. And so we've been amplifying our reach. We have a partnership with Combined Brain, and they cover about 120 different disorders, including ZTTK. And we're starting to work more with the children's hospitals and to some extent even oncology centers for the really complex cases. So that was Unravel as a whole, our approach. Now for ZTTK. So again, thanks to the really generous support of the ZTTK Foundation, we partnered with all of you to figure out how, what can we do for ZTTK? And just so you know, we knew nothing about ZTTK, right? Aaron and other researchers know far more than we do, and then there's all of you who know far more than any of us or the researchers will ever know. And so I think it's a very nice partnership to kind of complement the different skills and experiences. And so the purpose of the study was kind of threefold. So one is let's generate RNA data. So that's what I shared earlier, kind of that, what recipe do you want to build today? Let's get that information from patients and match controls. And that data set could be extremely valuable to all these researchers, getting other people to look at it using their AI tools or generate new ideas about what you could do with it. Let's get that data. If you don't have the data, you can't do anything, right? Then on our end at Unravel, let's use our platform, let's use BioNav to do this computational drug screening on a whole bunch of patients, and let's see what comes up. Again, we know nothing about ZTTK. We don't even put in ZTTK or the SON mutations or anything in our AI software. So this is just the RNA telling us what's disrupted and how can we fix it. And then the last part is, okay, let's narrow this list of potential drugs down for further investigation. You can't look at everything, there's just not amount of time or resources available. So what did we do? There were 22 sunshines and 22 controls. And so thank you to all of you. I mean, this is, again, it is your data, it is your experience that's driving these studies. Here are the results. So just to orient you of what's what. So this is a correlation plot, which basically means how similar is one patient to another patient. And similar not based on where the mutation is or age or anything like that, but based on all these 40,000 molecules. And again, not all of them are relevant, but it's probably a few hundred in the end. But out of the few hundred molecules, how similar would they be in terms of helping a given patient or even hurting a patient? So it depends on where they are on their list. And the more red it is, the more similar the patients are, and the more blue it is, the more opposite the patients are. And so similar, hopefully that's self-explanatory. The blue means, let's say aspirin is the top drug for patient 1, and there's a blue box between patient 1 and patient 2. Patient 2 would be predicted to be worse off if they took the same molecule, right? So they're kind of opposite in that sense. And this is across all these different drugs. So we can see very quickly that there are two groups. Now, we did a lot of analysis on the sex of the patients, the age, where the mutation is, all the stuff that you heard about today. Nothing lines up. Now, what is not shown here is all the data that you're contributing to both the citizen registry and the questions that are being asked. All of that will, I think, I've spot checked a couple patients that I've had a chance to talk with their families. I think that actually will clarify some of this grouping. But bottom line is the traditional metrics don't correlate. If you look at the SON gene expression, so this is how much of the RNA of the SON gene does a given patient have, in this case, relative to their usually parent, so same-sex parent. And so this is on a log2 fold change. Don't worry about what that means, but basically less or more, right? So less is on the red side, below that 0 line, and there's one patient who actually has more of the SON gene. Now, this is on the RNA level. It's like having maybe multiple copies of your recipe. You could still bake one cake with it, right? So we don't know what the protein is doing, and that's where Aaron's group at UAB was going to be looking at the protein side. But in our RNA analysis, yeah, one patient's actually higher than everybody else. But for the most part, the patients have less, and that's what we were expecting. But there's no pattern here that we were able to find. Nevertheless, there are these two groups. And hearing about some of the symptoms and some of the differences between patients, and some of these start lining up pretty well. And so again, I just want to stress this is for research use only. We just in general don't share drug names. We still have some categories for drugs here. This is not ready to go to the clinic by any means. This is just initial research. We haven't even finalized the report yet for the foundation. But I want to share this to show that there are these interesting categories, and there's potential for treatment. We'll have to see, to dig in a little bit more, but this is realistic. So in terms of the drugs that we predicted for all the patients, how do we help all ZTTK patients overall? There were like these thyroid drugs, certain anti-inflammatories, not all of them, by the way. A couple of these other molecular pathways. But then group 1 was very different from group 2, and as the colorings show. So group 1 had a lot more inflammation. Melatonin was interesting. So melatonin is tied to circadian rhythm, sleep. You might take it as like before bed to help you sleep. There's additional pathways related to that. Group 2 was all about metabolic function. And what we're seeing, and there's some data that I showed yesterday with the scientific advisory board, that some of the markers or genes that are behind these two groups actually do help explain why some of these drugs are coming up. Now, again, this is just a hypothesis. We have to test it. But the bottom line is that this one study led to this wealth of information. And it's not just theoretical. There are drugs behind all of these ideas, right? So if some of this gets confirmed, there is a path to work with clinicians, do studies, and get into the clinic very quickly. Again, with the support of the foundation, it has to be done rationally, think through the risks and benefits, but there is a path, right, if the data work out. And so just to conclude here, we generated data from the sunshines, right? So this data is available for researchers, for clinicians. It's there. If you don't generate it, you don't have it, it's hard to do anything. We predicted some very reasonable to translate, and by translate means getting it into the clinic, drug candidates, right? So these are all existing molecules. They're all available to clinicians for prescription. Now, they won't just prescribe them for no reason. There has to be a rationale, but those drugs exist. Many of them are quite safe, right? So there are some possibilities. And then what's in progress, and this is again, very near term that this will happen, is narrowing down those candidates based on some of the other data that I'm not showing. It gets very technical, but narrowing this down to a few different pathways that we want to validate. We want to do some additional work, especially with some of the fantastic researchers and clinicians that are here. But the idea is that from this one study, we have all these interesting directions that are very meaningful and impactful potentially. And so with that, I want to thank you for your attention. Again, thank you to those who participated, those who are continuing to participate and adding more data to our knowledge of ZTTK. And again, really excited to be here and happy to take any questions if there's time. So thank you.